Hi, Swetha.
Nice to meet you, by the way.
Good to meet you. Thanks for joining.
Of course, thank you for inviting me.
Oh yeah, of course. Do you want to test out your slides?
Yes, I just got started. Um,
So, so your volume got really much better just then. So I don't know if you were closer or what, but that whatever you're doing,
is
that's
is
good.
this better? I can just
Yes,
move.
yes,
Yeah.
yeah, that's
Okay.
good. Or do you have, or if you have a headset, but that right now it's good.
Okay. I'm gonna share screen and see if this works. Yeah.
Okay, and then try the slide view. Okay, looks good.
A couple of videos. I'm just gonna sort of make sure that plays well.
Okay. We couldn't have picked a better timing for you to give this talk.
I know, right? It's been a distracting morning or day, I guess.
I bet.
One more. Yeah, that played, right?
Yes, yes. Okay, looks good. Hi, Quinn.
Aaron,
Hi, it's Long.
that's better. Good.
Hello, hi.
Hey,
Quinn, do you want to test your
congratulations.
slides? Quinn, test out your slides if you can share your slides.
All right.
And then,
Full screen?
Yeah,
Could you see my screen or?
yeah, yes, yep.
Oh, but
And then
I
do
don't.
can you do the slide view
Slide
or the play?
okay.
Yeah, yeah, yeah.
Like this. Oops.
Did you hit play?
Yes. I don't know.
So
I
we
can't.
don't see.
You didn't see my slide or?
We can see your slides, but not in the slideshow view.
Oh, not in the slideshow.
Maybe stop.
Yeah.
Yeah, try to share again.
So weird. How about now?
Not yet.
Not yet. Okay. Let me try one more time. So maybe I.
Okay. Now go to slideshow.
Yes. Yep. How about now?
Wait. Oh, no.
No.
Oh, it's weird. Yeah, it's weird.
So
do you maybe quit PowerPoint and go back into it? Do you have more than one monitor or something?
No, I only have one monitor. Let me try.
All right. Maybe okay. Go to when you do share, rather than just sharing the PowerPoint, just share your desktop. But maybe
okay. Yeah. Let me try one more time. Sorry.
Okay.
So I tune the settings. How about now?
Yeah, that's good.
That's good. Okay. That's cool. Awesome.
Okay.
I want to try one more time. I want to switch my I want to use the laser pointer. How about now?
Yep, that's good.
Everything looks okay. Okay, good. Thank you. Now I stop sharing. Yeah. So who will be the first one?
You'll be the second one.
Oh, okay, that's cool. So the first one will be the piezo story.
Yeah.
So Swetha, I don't know. You know, a graduate student names Ming Huang is from China. It's rotated in your lab previously, and end up the Max Mo's lab in Scripps.
At Scripps.
Yeah, Scripps. Yeah, I don't know.
Okay. Yeah. Min? Did you say Min?
Min, yeah.
Yeah. I remember him. Yes.
He works 2 years in my lab and applied the graduate program to Scripps and got it. And rotated in the lab, but sadly end up in our lab.
Correct. No. I remember him because through my time at Scripps as well. I remember him being around in Florida at least.
It's sometimes 15 or something. Right.
Did you go anywhere, Zong, for the holiday?
No, I just go nearby. So it's mountain people say, so you really cannot go to anywhere supposed
Who
to
says
be.
that? Who says that?
Usually, every big holiday in China, you cannot go to the famous point like Hangzhou. Never, remember. So, never. So you will definitely regret. You were stuck on the traffic for hours and cannot do anything but waiting. So just go some nearby. Yeah.
I went to Chongming Island.
Do you get traffic? I heard a lot of traffic.
Yeah, a lot of traffic.
Okay.
But we had like a Buick van.
Oh, great,
Nice.
great.
Yeah.
Nice.
I think the height of luxury here is Buick minivan. It's really luxurious.
Yeah. We should get some RV sometime in China.
Oh yeah, I've been seeing those around.
All right.
Should we get started? Okay.
Sure.
Great. Okay, welcome everyone to another Neuro Zoom week. It's great to see everyone. One minor announcement for people watching this on YouTube, the YouTube live channel. I had to switch over to a different YouTube channel. I was just using one of my kids' YouTube's channels, and then YouTube changed their policy where if you're under 18, you can't, I think it's a good policy, you can't stream live videos. But anyway, I had to create our own. So now we have an official NeuroZoom YouTube live channel. So that's that. But you'll still see the link on the webpage. Everything else should be fine. Okay, so next week we have two great talks coming up. We're continuing, this is a month of Vollum Institute scientists, and next week we'll have James Frank, who'll talk about new chemical tools that he's been developing. And then we'll have what's sure to be a lively discussion about revisiting glia reprogramming in vivo from Chenli Zhang, long-time friend of NeuroZoom, and it'll be great to hear his latest results and talk about the implications. Okay, and with that, It's my great pleasure to introduce the first speaker today, Dr. Swetha Murthy, who is a scientist at the Vollum Institute. And Swetha earned her bachelor's degree in India at Bangalore University, and then came to do her PhD at SUNY Buffalo, working on mechanisms of NMDA receptor gating. Then came to the Scripps Institute, where she was a postdoctoral fellow with Artem Pataposhian. And here she studied the famous mechanosensitive ion channel piezo2, which was discovered as the channel that senses touch. And she, in a very elegant study using both gain of function and loss of function studies, showed that piezo2 can actually mediate sensitivity to pain, mechanical pain, using either optogenetic activation or conditional knockout studies. In addition to piezo2 studies, Swetha also discovered a family of other mechanically activated ion channels, the TMEM63 family, and showed that they were widespread in plant animals. She even has a new story where she found the mechanically activated ion channel responsible for carnivorous plants to open and close. So it's a really exciting field, so exciting that it was just today awarded the Nobel Prize. So it's great timing and great to have Swetha here presenting her latest research.
Thank you, Aaron. That was a very generous introduction. It's definitely been an exciting morning and day. I'm gonna get. All right, we're doing good?
Looks good.
Okay. Thank you. Welcome, everybody. Excited to talk about some of the research that I've done. The main story that I'm actually going to talk to you about is what I did in our DEMS lab, which is to evaluate the role of piezo2 in this phenomenon known as mechanical allodynia, or sensitized pain. And as Aaron mentioned, it was very fitting to see both David Julius and Ardem win the Nobel Prize today. So this is kind of an ode to sensory somatosensation, at least. So my lab is interested in studying mechanotransduction, which is the process of converting physical force into a biological signal. And turns out that our body experiences a wide range of physical stimuli. So the sensation of touch, which is a form of indentation, stretch, and shear stress are all physical forces that are sensed by cells and neurons in our body. And this conversion of physical force into a signal can be accomplished by receptors, ion channels, as well as intracellular proteins. But what my lab basically studies and what my interest has lied in is in mechanically activated ion channels. So these are channels that are membrane proteins, and they have something unique about them in a way that when there's force or tension applied to the membrane that surrounds them, the channel undergoes a conformational change and switches from a closed to an open state. This allows ions to flow through the cell. So if it were a neuronal cell and a cationic channel, for example, influx of sodium can cause depolarization and initiation of a signal. Non-neuronal cells, quite often calcium influx into the cell through a mechanochannel can then initiate a cascade of events. So it turns out this process is very important. It's for multiple physiological processes in the body. Some of the classical examples are our sensation of touch, where neurons that innervate the skin express mechanically activated ion channels and give us a sense of touch, acute mechanical pain, proprioception, deep pressure, and pleasant touch. We've known that there are mechanotransduction complexes that are present in these specialized hair cells that can sense frequency from sound waves and give us the ability of hearing. Blood flow in blood vessels can apply shear stress on endothelial cells that line blood vessels and can cause vasodilation or vasoconstriction. Another very obvious sort of physiological role for this is ocular pressure, right, which is very important to maintain both physiology and anatomy of the eye. And then a couple other examples are stomach stretch and urine voiding through bladder stretch. Now, although for the longest time it had been known that these processes depend on mechanotransduction, the sensor or the channel that was sort of responsible for many of these processes remained a mystery up until the recent identification of some of the eukaryotic mechanically activated ion channels. So just to give you some examples very quickly, we now know that piezo2 is required for touch and proprioception. TMCs, after much debate, is now known to be an essential component of the mechanotransduction complex in hair cells. And then piezo1 is also required for vascular development. However, there are still multiple sensors that the field collectively is still sort of looking for. And for those of you who attended Art Bader's talk a couple of weeks ago, he talked about how piezo2 in gut is required for gut sensation as well. So one of the main questions that sort of got me interested when I was a postdoc in our DEMS lab was we had this information that piezo2, or we kind of had conclusively started to figure out that piezo2 was required for touch sensation and proprioception. And this had been shown both in mice as well as Alex Chesler's group had shown that humans with an inactive variant for piezo2 also had lost the ability of discriminative touch and had severe proprioceptive deficits. But what had started to sort of puzzle us is whether piezo2 was required for acute mechanical pain sensation, or another phenomenon that was interesting was sensitized pain or mechanical allodynia. And we, at least the initial two publications that had come from our DEMS lab showing piezo2's role in touch, there was no pain phenotype that had been observed in the piezo2 conditional knockout mice. And Alex's group had done some quantitative sort of verbal assays with the human patients who had an inactive P2, but there was no quantitative pain analysis that had been done with these patients. So to test whether piezo2 was required for pain, acute pain or sensitized pain, we sort of got into the story that I'm going to talk about. So before I go into the data, I just wanted to sort of bring us all on the same page in terms of that mechanical allodynia or sensitized pain is actually one of the leading symptoms of clinical pain. And it's a process where innocuous touch causes a painful effect. So for example, after a sunburn, even the sense of the label from your t-shirt can cause pain. Turns out there are actually quite a few factors that can cause mechanical allodynia. So to name a few, chronic inflammation and nerve injury can cause sensitized pain. Diabetes patients quite often complain of chronic pain. And also chemotherapy drugs have been known to induce mechanical allodynia or sensitized pain. So despite the prevalence of this, the molecular mechanism or the sensor for the sensitized pain had not been identified or known for that matter. So the approach that I ended up taking was we took a piezo2, instead of using the piezo2 conditional knockout mouse, we used a sort of global knockout, but it was in a way, we kind of circumvented the idea that the global piezo2 knockout is lethal. So what we did was we crossed piezo2 floxed mice to a HOXP8 Cree mouse. And as you can see here on the left-hand side, HOXP8 expression is only in the caudal regions. So what this allowed us to do was to actually knock out piezo2 only in the caudal region. So the hind paw, tail, back, but then all of the respiratory, cardiac functions that piezo2 we knew was required for was kept intact. So this allowed the mice to be born and be healthy. Well, not healthy, but at least survive and facilitated our behavioral and other analysis. So to give you an example of how strong their proprioceptive deficits are, this is a video of a HOXP8 piezo2 knockout mouse. And as you can see that the hind limbs for this mouse is very uncoordinated. So the first thing we did was we wanted to recapitulate and make sure that at least touch was completely lost in these animals, and also wanted to make sure if we could identify a pain phenotype that we had not seen in the conditional knockout mice. So what you're seeing here is the percentage withdrawal from an animal when a Wanfrey filament is subjected to the hind paw of the animal. So the Wanfrey can apply specific amounts of force on the paw, and you basically note how quickly or how many times the mouse responds to that specific force. Just as a sort of a reminder, a force of 1G is considered as touch, and anything higher than that starts inducing sort of higher threshold, slightly pain-inducing stimulus. And what you can see here is while the piezo2 knockout, piezo2 wild-type mice started responding pretty well to forces anywhere between 0.4 to 4, the piezo2 knockout mice had completely abrogated responses when touched with as 5 as 4 Gs of mechanical stimulus. Now you can quantify this in a way that you can measure the mechanical threshold. So what's the threshold for a mouse to respond to a various or a range of physical force? And what we saw was that with the piezo2 knockout mice, there's a 5-fold higher threshold. So you need that much more pressure to get the animal to respond to a stimulus. So this is important because for the next few slides, I'm going to use mechanical threshold as basically an identification or as a mark to see how these mice respond to other paradigms. So sensitized pain, like I mentioned earlier, can be caused. There are two prevailing sort of models that has been suggested. One is inflammation-mediated sensitized pain, and the second is injury-mediated sensitized pain. So for inflammation-mediated sensitized pain, the very common paradigm that's been used in the field is to inject capsaicin in a specific region of the paw. And then after 5, 15, and 30 minutes of capsaicin injection, you can measure the mechanical threshold of mice in response to the capsaicin injection. So what we do is we measured mechanical threshold at baseline before injection, and then 5, 15, and 30 minutes after the injection. And what we see is that with the piezo2 wild-type mice, even 5 to 30 minutes after capsaicin injection, you see about an 80% reduction in the mechanical threshold, meaning now it requires very little force to actually cause the animal to respond. However, with the piezo2 knockout mice, they're kind of flatlined, right? So they see no distinction between before or after capsaicin injection. In the previous slide, I showed how there's a 5-fold difference in threshold. So just to make things easy, we've normalized both the conditions to among themselves. So what was actually pleasantly surprising and sort of revalidating our results in mice was Alex Chesler's group around the same time were doing similar studies. And the advantage that they had was they had access to some of these patients who had an inactive piezo2 variant in them. So what Alex's group did was they ran this pain assay on these patients, which is that, so they had a cohort of 4 control patients and 4 who had an inactive variant for P2. And the test that they tried was 2 3 mm by 3 mm, 3 centimeter by 3 centimeter regions are marked on their forearm. And out of the 2 regions, only one region is they apply a capsaicin patch on one of them. After 30 minutes, what they do is simultaneously on both the regions, A and B, they run a cotton swab on it. And then the patient is actually deprived of visual input, so they can't see what's going on. And what the patient is asked to do is simply say out of A and B, which stimulus, when they run the cotton swab, causes a painful sensitization or sense, basically. And what they noticed was with the control patients, 100% of the time, they can identify, even though they can't see it, they can identify which region caused sort of a painful stimulus by rubbing a cotton swab. However, the piezo2 loss of function patients can't make any distinction. And remember, these patients can't sense touch either, so they really don't even sense the cotton swab on their forearm. So every single time they were asked to guess sense, so there's like a 50% chance of getting it right out of A and B. So this kind of revalidated what we had seen in our piezo2 knockout mice. Now, because the other paradigm that we also went ahead and tested was that if during nerve injury, is the sensitized pain also dependent on piezo2? So again, we went back to our HOXP8 Cree piezo2 knockout mice. The paradigm that we used here is we measured mechanical threshold on day 0. It caused injury where it's an SNI. So both the tibial and the common peroneal nerve are ligated and sutured, and the sural nerve is kept intact. So all of the mechanical stimulus is applied on the sural side of the paw. And we then tested mechanical threshold both day 7, 14, and 21 after injury. And what we saw was same again with the wild type mice. Starting from day 7 to up to day 21, there's an 80% reduction in their mechanical threshold, which is an indication of sensitization. However, the piezo2 knockout mice actually don't have a much higher threshold. We do see that there's some compensation that starts occurring by day 14 to day 21, but we mostly think because these are such long durations, there could be other sort of intracellular gene regulation mechanisms that are probably causing them to recompensate for the sensitivity. So what both Alex Chesler's group and what my studies indicated was that piezo2 is the channel that mediates sensitized mechanical pain or mechanical allodynia. We think this can have profound clinical implications. And I think what was most thought-provoking in this case was that it was originally thought that pain pathways are not conserved between mice and humans. However, the fact that at least with the inflammation model, the fact that we were seeing similar results with the loss of piezo2, we believe that at least in this particular circumstance, the pathway is conserved, which becomes exciting for us because now we can sort of start targeting piezo2 as an important drug target to treat mechanical allodynia. And I'd like to note that last year, Bailong Zhao's lab actually came up with resolved the full complete structure of piezo2 as well, which I think will facilitate going forward sort of drug targeting for piezo2 and piezo1. So the one thing that we noticed, right, so far I talked about sensitized mechanical pain, but when I got started, I mentioned, I think one of the holy grail questions that are still remaining is there a sensor for acute mechanical pain? And what we had noticed was in our HOXP8 piezo2 knockout mice, if you apply blunt pressure as high as 400 G. Now remember, again, when we did the Van Frey test, our highest force was 4 Gs, whereas here we're going 100 times higher, right? So the way we run these tests are there are alligator clips that are sort of blunted off so that it's not causing any tissue damage to the animal. And by applying the blunt clip onto the tail of the mouse, we kind of try to see how quickly they respond to this stimulus. And what we noticed was with the piezo2 knockout mice, there was a minor distinction between the wild types and the knockouts, but they can still respond to pain. And this was the same case for the human patients as well, which is that they could still sense very high threshold pressure on the skin. Alternatively, or in addition to the behavioral assay, what we had also noticed was that if you isolate DRG neurons from the PSO2 knockout mice and mechanically stimulate the soma and record mechanically activated currents from them, there was still a response. So this assay, or the in vitro electrophysiology assay, is actually commonly used in the field. So just to sort of get us all on the same page, it's a whole cell patch clamp mode that the assay is done on. What you see here on the left is just a picture, it's a video, but it's a soma of the DRG neurons. Now these neurons are a bit unique, that although the protein's expressed at the periphery of the neurons, which is where the stimulus occurs, but they also express them on, the protein is also expressed on the soma. So you can culture DRG neuron soma and then record from them. And what you see here is a recording pipette on the right-hand side. This is the blunt probe or a stimulation probe on the left-hand side. And what the video shows is that you can apply sort of episodic time-regulated movements onto the soma, or indentation on the soma, that causes a mechanical stress on the membrane. And what you see here is just a representative trace of one of these currents from a DRG soma, where on top you have the micron, which indicates the amount of distance that the probe has traveled onto the membrane. And what you see here at the bottom is just the whole cell response from the cell. And you see that it coincides with when the stimulus is applied, and these currents basically inactivate based on their kinetics. So when you do this assay in wild type DRG neurons, you see that about 80% of these neurons do have a response, and there are about 15 to 20% that don't have a response. However, if you test this with the PSO2 HuxP8 knockout mice, although 40% of this responding neurons are lost, in the sense that they've now become non-responders because this was PSO2 mediated currents, there is still a population of these neurons that have a mechanically activated current in them, which sort of allows us to think, okay, which means that there is another sensor, mechanosensor, presumably required for acute pain in somatosensory neurons. So based on this result, we started screening and reevaluating some of the genes that we knew in the past and new candidates in the somatosensory in DRG neurons. And I came across this whole big family that's called as the OSCA TMEM63 family. So originally, OSCAs, which is the name of the gene in plants, were thought to be hyperosmolarity activated ion channels. But what we realized is that if you overexpress this gene in HEX cells and mechanically stimulate them, you do see mechanosensitive currents. So we think that the hyperosmolarity phenotype is actually sort of a side effect of mechanical stimulus, because when there's change in osmolarity, there's water influx through the membrane that can apply stress on the membrane. So just as a very quick introduction, this family's large plants have 15 genes. There's one in flies, and there are 3 genes in humans or in mammals. Since we identified them last year or early this year, Craig Montel's lab has shown that Drosophila TMEM63 is actually necessary or required for texture determination in flies. And what my lab now, since I've started at the Vollum, has been concentrating on are 2 major questions. One is to identify what are the sort of physiological roles of TMEM63s, mostly in mechanotransduction-dependent processes. And we're also using these plant genes, which are OSCAs, to start asking this very vital question of how, at the molecular level, how do these channels sense force? And I think the advantage of using the plant genes is that so far, most of the mechanosensitive channel families that we have known are usually 1 or 2 family members. So it's been a bit difficult to do comparative studies to see how different genes within the same family could confer mechanosensitivity. And I think the advantage that we have with OSCAs is that apart from the fact that they're 15, they all have very distinct biophysical properties. So the lab is now currently looking at what or how they could be responsible, or how do they sense force. And just so this is a very quick recap, like I said, so or if you look at just at the mammalian genes, there are three. What we see is that both TMEM63A and 63B, when overexpressed in HEX cells, do induce mechanosensitivity to these cells. So in this case, the top trace is the stimulus. The assay that we do is in the cell-attached patch clamp mode. You can apply negative pressure and stretch the membrane. And in lieu of the stretching of the membrane, you can record inward currents. So we've been able to do this both for TMEM63 and B, but with C, we haven't seen anything yet. So we're still unsure whether it is mechanosensitive or not. And the other point I would like to make is that their kinetics are very different from piezo's. So we're sort of curious to see how, especially in cell types that have both expression of TMEM63 and piezos, what role they could play. So sort of segueing from there, we currently have data suggesting that 63A and B is expressed in somatosensory neurons. So what you're seeing here are three different somatosensory ganglia: trigeminal, nodose, and dorsal root. On the left are just cross sections that are done in a lag-C reporter mouse, both for 63A and 63B. And on the right here for DRGs and in situ for 63A or B. And as you can see, they're both expressed. Interestingly, they're differentially expressed, meaning that the neurons that express 63A don't have B in them, and the ones that are enriched for B don't have A in them. So I think it's kind of interesting to see how these two are going to play out, if at all. It's also interesting to note that 63B is co-expressed in some population of piezo2 expressing neurons as well. So my lab is currently looking at how this interaction or how these two channels could play a role for mechanosensation. So with that, I'd like to thank you all for your attention, and also thank people who helped me accomplish a lot of the initial part of the studies. Ardem has been a fantastic mentor and advisor, so I was super happy for him today. Megan Loud, Kara Marshall, Ehab, and Elaine were three people who were monumental in like the initials and mechanical allodynia story. Adrian and Bill played a key role in the OSCA TMEM63 identification story. And it's been almost 2 months since I've started at the Vollum, and it's been a fantastic place, but currently small, but we're slowly growing. This is my lab here. So with that, I'm happy to take questions.
Thanks so much, Swetha, for the exciting talk, and we have time for questions. Zhao Xuan, you can ask.
Hey, great talk. So I'm wondering, when you, so can you completely abolish this mechanosensory current if you knock out both piezo and TMEM63?
So we're trying to do that, because we're trying to do a double knockout and potentially a triple knockout of 63A, B, and piezo2 to see if these mechanical currents are completely abolished or not. So yeah, we don't know that answer yet.
So is there any feedback to sort of pain and changes in expression of the channel, or change in its properties?
Yeah, so we don't have conclusive evidence, but we did think, or we do think that it is a possibility, right? especially, for example, with sensitized pain, one theory was that because of injury or inflammation, maybe there's overexpression of one type of mechanochannel and underexpression of the other one, right? That's why we were a bit pleasantly surprised to see that if you knock out PSO2, right, completely sensitized pain is completely gone. So I think the short answer, there's no conclusive evidence yet, but there is there could be a possibility. Also, I think I'd like to add that because it's such an important role, both touch and pain, that maybe there are multiple channels to compensate for these phenomena if something happens to one of them.
Got it. Got it. Great. Any other questions? Okay, thanks so much, Swetha. Good luck with your new lab. Looking forward to future discoveries. And
Thank you.
Zilong,
Aaron and Zilong.
Okay, great, thanks, Swetha. Okay, it's my really great pleasure to introduce Dr. Kun Li from Tsinghua University. So Kun has been one of the brightest Iowan graduates of history. So Kun had her bachelor degree from Jilin University, and after the bachelor degree, she went to Professor Hailen Hu's lab. Back then, it was just downstairs of our lab in Iowan. So when Kun in Hailen's lab, they make an amazing discovery to find out the critical role of beta MK2 in habenula, so getting the neuroactivity, also having an important role to regulating the depression-like behavior in mouse. All the amazing story end up the beautiful science paper. So after PhD, Kun went to postdoc with Ned Hens and Rocky Rockefeller, and there Kun make quick discovery to find out there's sexual dimorphic circuitry regulating the oxytocin dependent emotional behavior. So from 2020, Kun come back to China and start her own lab in Tsinghua, also in the McGovern Brain Institute, and started her own lab in PI, starting focusing on the sexual dimorphic neurocircuitry again, not only in emotional behavior, but also, I believe, in a series of psychiatric disorders. So today, Kun will talk about her recent unpublished story about a cortical-hypothalamic circuit for sexual dimorphic dependent sociosexual behavior. Okay, welcome, Kun. She was on the line a minute. Okay, here you go.
Okay, thanks very much for those nice introduction, and the time in IO is really memorable for me. So now I would like to share my screen. Okay, so could you see my screen? Okay.
Yeah, go ahead.
Okay, sure. So thanks very much to Aaron and Zilong for offering me this opportunity, and I'm very glad to introduce our unpublished work on the sexual dimorphic neuromechanism of social sexual behavior. And male and females of many species look dramatically different from each other, and the sexual dimorphism can express in many ways, such as shape, size, and color, and even behaviors. And in the past decade, the neuromechanism underlying sexual dimorphism in social behaviors has been well characterized by many excellent works from David Anderson's lab, Neuro lab, Catherine Dulac lab, and many other labs, and as well as Xiaopeng Xu's lab from Iowa in China. So previous research on sex difference focused on the hypothalamic function in social behaviors. However, we know very little about whether other brain regions other than hypothalamus can sexually dimorphic regulate social and emotional behaviors. Okay, sorry. In the In the past, in human, gender differences are common in the incidence of many mental disorders. And the research in clinical study has suggested that females are more susceptible to depression, anxiety, whereas males have higher risk for alcohol and drug abuse and childhood ADHD. Recent transcriptomic data from human brain samples reveals that male and female forebrain show a very low overlap of differentially expressed genes in both depression and PTSD. So this result suggests the gender differences do exist in the prefrontal cortex. And the sex-specific susceptible and the resilient mechanism in the prefrontal cortex may contribute to the gender differences in mental disorders. And it first implies that the cortex may encode normal social and emotional behaviors by genders, or both genders by sexually dimorphic mechanism. So when I started my lab, the question we asked is, how do sex differences in molecules, cell types, and circuits lead to gender differences in social and emotional behaviors? And the brain region we are looking at is the prefrontal cortex. Sex hormones are essential to cause gender differences. At the developmental stage, the major function of hormone is to shape the structural differences between sex. While in adulthood, sex hormone could generate sexually dimorphic behaviors by activating the specific neurocircuitry in the brain. For example, the estrogen level in males are correlated with their hierarchical relationships in social groups. And while in females, the female will develop this estrous cycle because of the periodic fluctuation in ovarian hormones. And it's very difficult for me to go to the next slide. Okay, cool. Female estrous cycle can be divided into 4 stages. The proestrus, estrus, metaestrus, and diestrus. And these 4 phases cycle every 4 to 6 days. And the female animals will become sexually receptive when they are in estrus. And we can quickly identify these 4 stages by the vaginal cytology. So it has been reported that the estrous cycle has dramatic impact on behavior response and the neural activation. So this work shown here, when exposing female rats to TMT and innate fear-inducing odor, female in estrus will get higher fear response to TMT, and their brain region will be significantly increased, activated in many brain areas as compared to the other phases of cycle. And another very interesting work from Neuro Shine group has shown that the estrous cycle can regulate both the structure and the synaptic plasticity of the projections from VMH to anterior ventral hypothalamic nucleus, AVPV. Moreover, this connection could link the female sexual behavior to estrous cycle. So all this data suggests that the female's hormone status is correlated with the alteration in both neural activity and the behaviors. So oxytocin is a homopeptide important for social behaviors. So it is released in the hypothalamus and acts through the oxytocin receptors, which are widely expressed in the whole brain. So Miho Nakajima in my post lab has identified a specific class of oxytocin receptor expressed interunit neurons in the mouse medial prefrontal cortex. So I first validate the oxytocin receptor expression in different cell types in the MPFC. So, and we found 42% of the OSTR are somatostatin positive, while 50% of the OSTR are excitatory. And importantly, the enrichment of OSTR in are much higher than that in VGLUT1 positive cells. So this result suggesting a primary inhibitory effect of OSTR inhibition, OSTR function on the local circuit of MPFC. So next, we will generate a back mouse line to first characterize the function of these cell types. And she observed females with OSTR interneuron inhibition show much less time in sniffing males during estrus phase. However, the social sexual behavior in males was not affected by OSTR interneuron inhibition. And in the follow-up work I did in my post-lab, we reported the sexually dimorphic post-synaptic response of OSTR interneuron activation. And interestingly, I would like to point it here. In females, oxytocin receptor interneuron activation have a major inhibitory effect on layer 5 pyramidal neurons. So despite the sexually dimorphic function of OSTR interneurons in the MKFC, but the neural mechanism for estrus-dependent sociosexual behavior in females remains unknown. So my student measured the RNA level of OSTR in the MKFC, and we didn't see any change of OSTR with the different estrus cycles. So based on this result, we hypothesized that so the other neurons in the OSTR connected circuits would respond to estrus. So because the layer 5 pyramidal cell are very heterogeneous population with different output. So it's important to know which subpopulation of layer 5 pyramidal neurons receive direct synaptic input from OSTR interneurons and regulate estrus-dependent sociosexual behavior in females. To find out this subpopulation, we use a crate-dependent HSV virus, which was developed by David Anderson lab. We express this virus in oxytocin receptor crate mouse and observe the downstream signals in different other brain regions. So interesting, we observe the virus labeling cells in the anterior hypothalamic nucleus. Because the polysynaptic labeling of this virus, so we first confirm the monosynaptic connection of OSTR and the agent projecting layer 5 cells by CTB. So these red cells shown in this picture are layer 5 pyramidal neurons project to HN. So next we examine the synaptic connection, the monosynaptic connection between OSTR interneurons and the agent projecting layer 5 pyramidal cells. So we found that the probability of monosynaptic connection between these two populations is around more than 87%. And interestingly, the inhibitory effect of OSTR inhibition on agent projecting neurons is stronger, much stronger in females than males. So which is also consistent with the result reported in our previous paper. So based on this evidence above, we are very confident to conclude that the agent projecting layer 5 neurons receive monosynaptic connection from oxytocin receptor interneurons in the MPFC. So I'm particularly interested in the MPFC agent pathway because agent is part of the sexually dimorphic nucleus complex. Even more interestingly, age lesion could damage sexually behavior specific in female rats without affecting the male sexual behavior. So therefore, so we think that maybe the MPFC agent pathway may regulate the sexually dimorphic social sexual behavior in females. So to test this, we first measure the synaptic plasticity of this pathway in both men and females. Very interesting, we are very surprised to observe increased spontaneous frequency and amplitude in this in the estrus females, are not in the diestrus and males. And this evidence suggests that the synaptic plasticity of the MPFC agent pathway are changes with estrus cycle. And in estrus phase, not diestrus phase, the synaptic strength of this pathway is potentiate. So next, we measure the membrane potential in these agent projecting neurons in both sexes. And we found that the agent projecting neurons are more hyperpolarized in these estrus females. And first of all, oxytocin application could decrease the resting membrane potential of these neurons in the estrus females. So again, this result suggests that oxytocin receptor internal activation produce a primary inhibitory effect on H projecting their 5 pyramidal cells. So this result looks like a contradiction with the increased synaptic transmission of H projecting neurons during the estrus stage. So to further explore the potential molecular mechanism leading to this contradiction, so we use a translating ribosome affinity purification approach, which we call the TRAP, and this method is developed in my post lab. So we use TRAP to profile all the translating mRNA in these H projecting neurons. So specifically, this method is that H neurons were infected with the retrograde AV virus expressing JP-tagged ribosome protein L10A. And 3 weeks later, we could nicely label a subpopulation of these layer 5 pyramidal neurons in the MPFC. And next, we immunoprecipitate the translating mRNA in these cells with a EGFP antibody. And then we do the RNA sequencing to characterize the translating molecular profiles of this subpopulation. So by comparing the input and IP samples, and we have identified source of significant genes enriched in this population. And these red cells define the location of H projecting neurons in the MPFC. And this image shows expression pattern of identified genes enriched in this pathway. So the expression pattern of these genes in MPFC are quite similar with the location of H projecting neurons labeled by CTB, suggesting the high specificity of this pathway-specific trap method. So based on our slides recording, we use the following 3 criteria to screen our candidate. First, it should be activated by hyperpolarization. Second, it can increase the spontaneous EPSC. And third, it could regulate the social behavior. Among the gene list, there was one candidate caught my attention. It is CACNA1H. So CACNA1H encodes T-type calcium channel CAPS.2, and it is mainly expressed in the layer 5 pyramidal cells in the MPFC, and it has also been shown that hyperpolarization could increase the synaptic transmission in cells express CACNA1H, and this effect can be blocked by T-type channel blocker and the CACNA1H deletion. Moreover, a mismutation of CACNA1H gene has been reported in autism patients. So according to our AFI state and the reported function of CACNA1H, so we hypothesize that the CACNA1H may play a role in synaptic plasticity of the H projecting neurons during estrus. We tested this hypothesis in vivo. So we express a Cre-dependent fluorescent protein in CACNA1H Cre mouse line to specifically label these neurons in the MPFC. So by performing the whole cell recording, we found that in estrus females, the CACNA1H positive neurons show significantly increased spontaneous EPSC frequency compared to the CACNA1H negative cells. So this data is also consistent with what we observed when we recording the H projecting neurons. So we know from this result that the estrus cycle can regulate the neural activity of CACNA1H neurons in the MPFC. So the next question we asked is whether CACNA1H regulates the sociosexual behavior in vivo. So to assess this possibility in vivo, we expressed shRNA to knock down CACNA1H in the MPFC and test animals' behavior. So after the behavior test, in the vaginal smear of individual female will be collected to determine the estrus cycle. In female mice, CACNA1H knockdown could reduce their social preference of males only at the estrus stage, but not the other non-estrus stages. So unlike females, knocking down CACNA1H in males tend to increase the time that males sniff females. So because of the limitation of knockdown efficiency, my lab generate a mouse line with flocked alleles of the CACNA1H gene. And we found CACNA1H knockout in the MPFC significantly increase the social preference of male mice toward females. And while in females, CACNA1H knockdown decreased sociosexual behavior specifically at the estrus phase of the cycle. So based on all these two behavior experiments, so we can conclude that the CACNA1H in the MPFC have the sexual dimorphic role in regulating sociosexual behaviors. And more importantly, its function in females is estrus-dependent. To first examine the psychodimorphic mechanism of sexual behavior at a cellular level, we use a chemogenetic approach to specifically inactivity cells in the MPFC. And we found that CACNA1H inactivation produced consistent behavior results with reduced expression level of CACNA1H gene. And the activation of CACNA1H neurons is required for the sociosexual behavior in females during estrus phase. In order to get the insight into the molecular mechanism regarding the sexually dimorphic role of CACNA1H neuron in social behavior, so we systematically compare the translating profile of CACNA1H neurons of females at the different estrus cycles using trap method. So by hierarchical cluster analysis of gene expression pattern, we found 5 groups of genes whose expression fluctuate significantly across the estrus cycle. But the expression level of this, but the very few differentially expressed genes between male and females are observed in these CACNA1H neurons, suggesting that gene expression of in CACNA1H neurons responsive to estrus cycle. So more interestingly, the CACNA1H gene belongs to the class 3. And we found that this gene reached its highest expression level in estrus phase and decreased in other phase of the cycle. So we further validate the RNA sequencing data by detecting the CACNA1H mRNA with both RNA-scope and real-time PCR methods. And we found the significant upregulation of CACNA1H mRNA in estrus females. So given the fluctuation of CACNA1H expression, so we first recorded the T-type calcium current of CACNA1H in CACNA1H neurons. And we found that the amplitude of the T-type current in the MPFC of CACNA1H neurons significantly increased in the estrus females. So this data provide further evidence for linking the CACNA1H function to estrus. So to further characterize the activity of CACNA1H neurons in vivo during sociosexual behavior, we use fiber photometry system to record the calcium activity of these CACNA1H neurons in freely moving animals when they perform in the sociosexual behavior. And interestingly, we found that the calcium signal from CACNA1H neurons decreased significantly before the onset of each sniffing of males, and then increased gradually until peaking after socialization with males in these estrus female mice. And this pattern of calcium signal that drop first and then increase in CACNA1H neurons is unique to estrus females. And we didn't observe this kind of activity pattern in both the estrus females and in males. So we think this data indicates that the inhibition of presynaptic OSTR interneurons in estrus females will allow more capsicum to channel to recover from inactivation and subsequently open, resulting in substantial CACNA1H neuron activity changes, and which encode the social sexual behavior in estrus females. So our last experiment is to elucidate the role of the MPFC AHM pathway in the in social sexual behavior. So we express a a dread virus in capital 1H neurons and infuse Tsinghua in the AHN via cannula. So we found that inhibition of capital 1H exon terminals in the AHN could significantly decrease the social behavior in estrus and increase social preference in males. And this result suggests that MPFC-AHN pathway is a direct downstream circuit that mediates the sexually dimorphic and estrus-dependent role of OSTR interneurons in social sexual behavior. So finally, Finally, I would like to summarize our work in this model. So we found in estrus female, CACNO1H is enriched in MPFC-AHN pathway is significantly upregulated. And the release of oxytocin, reactive oxytocin receptor interneurons, resulting in the hyperpolarization of postsynaptic CACNO1H neurons. And this kind of inhibition from presynaptic OSTR interneurons will restore and open more calcium to channels on CACNO1H neurons and significantly increase the synaptic activity of this MPFC-AHN pathway, thus producing the social approach behavior in estrus females. So I think in this work, we have identified the CACNO1H express neurons receive the direct monosynaptic input from OSTR interneurons in MPFC. And we have uncovered a novel neural population of the MPFC and its downstream pathways that respond to the periodic changes of the estrus cycle in females. And we also demonstrate a sexual dimorphic neural mechanism underlying estrus-dependent social sexual behavior at the molecular, cellular, and circuit level. So finally, I would like to thank all the people who make this work possible. So first, I would like to thank my postdoc mentor, Knight and Ines. So I have identified the CACNO1H when I was a postdoc in his lab. And my students, Yuping and Xiaomao, did the behavior study and the did the slides recording. And Naku helped us with the photometry setup. Then and Lipson provide us the CACNO1H cream mouse. Okay, and thanks very much for your attention, and I'm very glad to take any questions.
Great. Thanks for the main talk. I have a quick question regarding the last piece of data you showed. Since blocking the from CACNO1H to AHN has increased social sexual behavior in male. Why is that?
Yes.
So do we have any explanation? Yeah. Why is it increased in male?
Yeah. So this is another ongoing project in my lab. So we think maybe the local circular connection is quite different between the male and the females. So we mapping both the input and outputs of CACNO1H in male and females. We think that maybe the different input and output circuits mediate the different function of CACNO1H in males.
Okay.
Yeah.
All right. Any more questions? Yeah. I'm going to throw another quick question about the perspective autism connection about the CACNO1H mutations, right? So it's quite interesting, but yeah, I don't know. But in terms of general sexual behavior, so ASD patient seems fine, right? So you have normal drive, normal. So I don't know. Do you have a particular insight about how the not social sexual behavior, but in terms of general social interaction behavior, maybe it's a specific population of the PFC neuron might be involved with some another nucleus or the HN to regulating the social interaction behavior, right?
Yes. Yes, we also look at the normal social interaction behavior between the same sex, and we didn't see this kind of sexual dimorphism. So this sexual dimorphism is specifically to the sociosexual behavior. And we also look at the other emotional behaviors, and we have some our preliminary data suggests that it can also regulate sexual dimorphically regulate the emotional behaviors between male and females. Yeah.
Okay. Or this interesting gene might express somewhere else, right? Not
Yes.
only in the Pepsi,
Yes,
right?
yes.
So,
Yes.
so
the manipulation we did is specifically in the medial prefrontal cortex.
okay.
And
Okay.
we also look, actually, when I do the iron scope between estrus and diestrus, we also look at the other cortical regions. So we see a slight increase of this gene of in other brain regions of estrus females. Yeah.
Okay. So one last question. So, for expression of the gene might be regulated by some hormone, maybe in the cortical pyramid, right?
Yes, yes. So we in our trap-seq data, we look at all the enriched genes in this H neuron, and we found the G-protein carboxyl receptors was expressed in cells. So we think maybe this is a reason why it can be regulated by the estrus cycle. Yeah.
Makes sense. Great.
Yeah.
Right. So people seem still largely in a holiday mode. Is there any more questions?
Hi, Kun.
Hi, please.
你直把声音听。So I'm interested. What might be the functional roles of the oxytocin receptor interneurons in prefrontal cortex? Because you show that during the estrus period, the EPSC, right, was increased.
Yeah.
But in your last slide about the model, actually the oxytocin receptor interneurons are involved, right?
Yes, yes, yeah, yeah. Maybe I didn't explain clearly here. So we think that the expression of PACN1H, because this gene encodes the calcium 2 channel, it is a hyperpolarization activated T-type channel. So the presynaptic inhibition from OST interneurons will activate this channel and restore and make more recovery of these T-type channels, and then make and then resulting the increased activity of these cells. So which we think the increased transmission of this pathway will encode the sociosexual behavior.
So it's like a sort of rebound excitation.
Yes, it's similar like that. We think the presynaptic inhibition will amplify the T-type calcium current. Yeah.
Okay, thank you.
All right. Any more questions? Okay, it's no more questions. Thank Swetha and Kun for great talks today.
Okay, thanks.
Thank you. Thanks, Swetha. Thank you.
Yeah, for this opportunity. Yeah.
Thanks.
Great.
Great talk.
Thanks,
Good
everyone.
to see everyone.
See you all next week. Yeah.
Bye, guys.